Today we are going to be talking about the effects of mutations in your DNA. Specifically, we are going to be looking at point mutations. Point mutations describe what happens when one base is substituted for another. When the mutated DNA is transcribed into mRNA, the mutation may affect the transcription of the mRNA into its protein counterpart in a couple of different ways, or none at all. It is possible that the substitution of one of the nucleotides for another may not affect the protein at all. In this case, it is called a silent mutation, as the mutation doesn't affect the gene product. This is possible because some amino acids on the tRNA have multiple possible anticodons that match to multiple codons in the mRNA. So sometimes a base can be switched out for another one that results in the same amino acid. Another possibility is that the substituted base does result in a change in the codon that results in a different amino acid. These mutations that cause changes in amino acids that may alter protein function are called missense mutations. There are also a couple of special instances in which the amino acid that is changed is either changed to a stop codon, called nonsense mutation, that results in a premature stop to the amino acid chain, or another possibility is that an existing codon that codes for a stop codon is changed instead to code for a functional amino acid, making the protein continue to grow instead of stopping. This is called a read-through mutation. Another couple of important vocabulary terms are transition and transversion. A transition describes when a purine base is converted to another purine base, while a transversion details the exchange of a pyrimidine for a purine or a purine for a pyrimidine. Now we are going to look at some examples of these mutations. Here is our DNA coding strand. First, we are going to transcribe it into our mRNA, which means just changing out the T's for U since this is the coding strand. Then we are going to translate the codons into amino acids using a coding chart, which I've already done here for the sake of time. Now we're going to look at all of these different mutations in the DNA and the effects they have on the mRNA and ultimately the amino acid sequences. In this first example, we have a T to C pyrimidine-pyrimidine transition. The result in the amino acid is it's an alanine, which is what it already was. So this is a silent mutation since there's not a change in the protein product. In this next example, we have a C to A pyrimidine to purine transversion. This results in a lysine, whereas before it was different. So this is going to be a missense mutation. Here we have another C to A transversion. This results in a premature stop codon, so that is going to be a nonsense mutation. And in this last example, we have an A to G purine-purine transition. This is going to result in an amino acid, whereas before it was a stop codon. So this is going to lead to a read-through